Myself, Dinesh Kalra. I am from Dayanand Public School, Pansar Nabha. In this video, I will discuss about search for the genetic material from the chapter Molecular Basis of the Inheritance. We know that genetic material helps in the transmission of the characters. During the mid-19th century, it was known that chromosomes are made up of DNA and the proteins. But out of these, which molecule is responsible for the inheritance was unknown. So, various experiments were done to prove this. First of all, Frederick Griffith in 1928 proposed transformation experiment. He worked on Streptococcus pneumoniae. There are two strains of this bacteria. One is the smooth strain. This strain has a mucus coat and so it is a virulent. That is, it causes the disease. Second is the rough strain. This strain lacks mucus coat, so it is the avirulent. That is, it does not cause the disease. He performed his experiment in the various steps. First of all, he injected S strain to the mice and then found that mouse dies. In the next strain, he injected R strain to the mice and mice was the live. In the third set, what he did, he heat shock S strain and then injected to the mice, but mouse was the live. In the last step, he mixed R strain and the dead S strain, then injected both these into the mice, and to his surprise, he found that mouse dies. From this, he concluded that there is something transforming principle which has transformed R strain to the S strain. But he could not explain about the biochemical nature of transforming principle. That is, whether it is DNA, RNA or the protein. Then later on, three scientists, Avery, MacLeod and the McCarthy, they continued his experiment. What they did, they took heat shock S strain and divide into the three samples. In the one test tube, they added proteinase, means in this test tube, protein was inactive. In the second step, they added RNase, means there is no RNA. In the third step, they added DNase, means there is no DNA. Then they mixed them separately with the R strain and then injected into the mice. They found that in the first case and the second case, transformation occur, but in the third case, there was no transformation. This shows that there is the DNA which help in the transformation. Then later on, Hershey and the Chase worked on the E. coli and the bacteriophage. Bacteriophage, the viruses that attack on the bacteria. What he did, they separately cultivated bacteriophage in the two media. In the one media, they took the radioactive labeled S, means in this media, the bacteriophage which grow, they have the protein which is a radioactive. In the second set, they use radioactive phosphorus. This shows that in their DNA, there is the radioactivity. Then they injected both these strain into the E. coli. Bacteriophage multiply in the E. coli. Then ultimately they centrifuge these culture and they found that there is no radioactivity detected in the cell of the first set, but it was present in the supernatant. And in this case, they found radioactivity in the cells and no radioactivity in the supernatant. From this, they concluded that the sample in which DNA was present shows the radioactivity, and from this they show that DNA is the genetic material. Now let us find What are the properties of genetic material? Means what are the properties of the DNA? It should be able to replicate. It should chemically and structurally stable. It can show the mutation. It should be able to express itself. It should be more stable as compared to RNA. RNA is used as a genetic material as well as a catalyst and the more reactive. But it has been shown that DNA has evolved from the RNA. So in this we have discovered that DNA is the genetic material. Thank you.